Hello everyone, my name is Vijay. Today we are discussing about a topic known as the kinematics of the double helix unwinding. It's all about like the timing or the motion and the factors that affect the unwinding of the double helix DNA. The first of all is the temperature. High temperature provide more energy for like breaking the hydrogen bond present in the base pair so that the unwinding get faster. The second is the denaturation, often achieved at the high temperature like 95 degrees Celsius and above to separate the DNA strands completely. And the third is the pH, which is about 7. It's optimal for the maintaining the stability of the double helix and the facultating control to the unwinding. Extreme pH can be disturb the hydrogen bond, lead to the denaturation and the instability of the unwinding the DNA. Third is the ionic strength. The ionic strength is like the salt concentration can seal the negatively charged phosphate backbone which is present in the DNA, stabilizing the double helix and make it more resistance to unwinding. Mm, like low salt concentration can uh, destabilize the double helix and make it easier to unwind. And the next is the DNA sequence. The DNA sequence in which is known as the glycine cytosine region. It is high rich in concentration and it have like three triple bonds between it, so it make mm, the DNA very stable and contain more uh, guanine and cytosine base pair, which are three hydrogen bond. Adenine and thymine also are present there, but they have two hydrogen bond. These make them more stable and resistant to the unwinding. AT rich region, also known as the adenine thymine region, easier to unwinding due to the weak hydrogen bond as compared to like the guanine and cytosine pair, as they have the three hydrogen bond between them and they make more stable for like effect from the temperature, denaturation, pH, ionic strength, and other things. Now come under the proteins, uh, the helicase enzyme that use ATPase hydrolysis to break the hydrogen bond between the base pairs. Uh, Actively unwind the double helix. Single-stranded binding protein SSBs bind to a single-stranded DNA, preventing it from the re-annealing and the maintaining the separate strands. Mm, this is all about the kinematics of the double helix unwinding DNA, and like also it is first studies by the Kung in 1957, and unwinding occurred due to the disruption in the interchain bondings. And also a theory come in between this as the rotational Brownian motion theory. It is the random change in the orientation of the polar molecule due to the collision of the other molecules. Here, the Kong studied that the DNA molecule which have molecular weight of 3 into 10 raised to power 6, which have 450 tons, take 150 days according to the theory. But in the real, if a DNA molecule having 10 raised to power 8 molecular weight, it can turns into 1 minute in the unwinding. It takes 1 minute in the unwinding. Now the second portion come here, the interaction of the double helix with the small molecules and the ion. uh, Here is the like first of all the base pairing interaction, hydrogen bond with the AT and GC base pairs holds the double helix together. And the second is the groove interactions, major and the minor grooves provide binding sites for the protein and the small molecules. When DNA get like in the transcription translation, in these process these group are much more helpful during these reactions. And there is the phosphate backbone interaction. As we see that the DNA is double helix, and inter in the inner side they are like guanine, cytosine. uh, they form the bonds, like three bonds of the hydrogen, but in the back bonding there are the phosphate which provide the back bonding to the DNA. The electrostatic interaction with the ions and the proteins stabilize the double helix. Like as we know that the interaction with ion and protein they stabilize the like double helix, but if the different ion came there and they can easily disrupt the uh, like the back bonding of the double helix DNA, and that can destroy or denature or we can say unwind the DNA. The fourth one is the intercalation. Intercalation agent insert between the base pair disrupt the DNA strength. Like they intercalations are agents that break the interlinked bond which is present there. The covalent modification, uh, like methylation and other modification, can alter the DNA strength and the function. These are different like ions which can alter the uh, or denaturation the DNA. Mm. After that, the sixth point is the DNA damage. Reaction oxygen species can the reactive oxygen species can cause the damage to the DNA. And the protein binding, transcription factor and other protein binding to the DNA are regulated by the transcription. We have here are the 7 points which can like we are telling about in this like interaction of the double helix in the small molecules and the ion. The first is the base pair interaction, second is the groove interaction, the third is the phosphate back bonding interaction, the fourth one is the intercalation, the fifth is the covalent modification, sixth is the DNA damaging, and the seventh and last is the protein binding. These all small and can interact with the double helix and can alter or change the DNA and can lead to the unwinding of the DNA. That's all for the video. I covered my topic. Thank you very much. Have a nice day.